Hello, welcome everybody to the Protists Reference Genomes Symposium at the Biodiversity Genomics 2024. I am Javier del Campo, a group leader at the Institute of Evolutionary Biology in Barcelona, and I'm going to be the chair of the symposium. This symposium has been sponsored by the Catalan Initiative for the Earth Biogenome Project, relatively small initiative based in Catalonia that has been part of the EBP since 2018, and we are working with endemic species of our region. And the title of this symposium, the complete title is Protist Reference Genomes, Gaps, Challenges, and Opportunities. And why? Because protists, if you don't know, they are all those eukaryotes that are not animals, plants, or fungi, which represent most of the genomic diversity of the eukaryotes. And they are not easy to work with. The first time I heard about the Earth Biogenome Project and the intention of sequencing all the eukaryotic life on Earth, the first question I asked was, really? All of it? They say, yes, all animal, plants, and fungi. I said, okay, and what about protists? And now many people, they are finally involved in the effort of generating reference genomes for protists. But what makes protists special? Well, they are special because they are very, very abundant and they are very, very diverse. There are 10 million predicted protist species, but there are only 80,000 described protist species. Among those, we only have around 3,000, 4,000 protist cultures available. And those are the organisms that we most likely will be able to use to generate genomes. And nowadays, we have less than 500 protist genomes available that we can use for our daily research. Why we don't have more genomes for protists? First of all, because they are not charismatic organisms such as animals, plants, or fungi. And second, because, as I said before, they are not easy to work with. Protists, the size of the protists and genomes is span from 0.55 megabases to 600,000 megabases. That covers a huge range of sizes. And all the other organisms that you can imagine, they will fit within these ranges as well. So they have diverse genomes, not only in terms of size, but also in terms of how they are arranged. And that makes complicated working with them. As well, the methods that we have to generate reference genomes are not properly adapted for protists. They have been designed for other kinds of organisms, such as mammals or vertebrates. And they don't have a lot of DNA, which complicates even more generating these persistent genomes. Luckily, today, we have with us 4 scientists that they have been courageous enough to accept the challenge. Guifré Torruella from the Institute of Evolutionary Biology, Ramon Massana from the Institut de Ciències del Mar, both in Barcelona, and Ori Francis and Amjad Khalaf from the Wellcome Sanger Institute. They will talk about their experience and expertise exploring the amazing genomic diversity of protists. Thank you all. The floor is yours.
Hello, I'm Guifré Torruella, and during my previous postdoc at the Comparative Genomics group of Toni Gabaldon in Barcelona, I worked on the genome project of apusomonads, in particular of the species Syngechia montserratensis. First of all, I want to thank my colleagues and the funding agencies for making this work possible. We have a problem in genomics diversity. We have an unbalanced situation for free-living protists. As we can see in this phylogeny, all multicellular eukaryotes originated from unicellular ancestors. So in order to understand the origin and evolution of the eukaryotic cell, its cell biology, we need to sequence and compare the genomes of free-living protists. In this phylogeny, on top of each lineage, there are the numbers of available genomes and transcriptomes in red and green bars. About 90% of sequenced eukaryotic genomes belong to multicellular lineages, such animals, plants, and fungi, arrows in pink. About 9% are parasites in yellow. 1% are free-living protists, but mostly algae in orange. And just less than a dozen genomes are available from heterotrophic flagellates with key phylogenetic positions to understand the early evolutionary history of eukaryotes. This is why consortia such the Catalan Biogenome Project within the Earth Biogenome Project or the Darwin Tree of Life, to mention a few, are fundamental initiatives to overcome this unbalanced situation. Over the past decade, we have learned that heterotrophic biflagellates are widespread along the tree of eukaryotes. And today, I will talk about one of these lineages, the apusomonads. Apusomonads, here in red, have an important phylogenetic position. They are the sister lineage to Opisthocomta, which contains animals, fungi, and their unicellular relatives. So far, there is only one apusomonad genome available for the species Thicamonas. So, if we want to reconstruct the last common ancestor of these two eukaryotic groups and tackle certain evolutionary questions, we need to sequence the diversity of apusomonads. Currently, we are sequencing the genome of other species of apusomonads, and today I will focus on the genome of Syngechia montserratensis, also known as Monse. For that, we have sequenced Monse in 3 different conditions. The first one was the monoeukaryotic culture containing several natural bacteria that feed Monse. Since 75% of the reads were prokaryotic, we tried to reduce the bacteria by feeding Monse with only one sequence bacterium, without any other nutrients. But this approach was not successful. This is why we filtered the culture by 0.8 microns, which substantially reduced the bacterial load. Overall, what we are facing is a low complexity metagenomics issue, in which we have to untangle the eukaryotic genome from the rest. Here you have the assembly pipeline that we have designed. Starting from the top, we feed the sequencing data from the 3 different conditions. We apply standard filters to the reads, and we assemble them using metaspades to obtain the primary assembly. From that, we ensure that there is only one eukaryotic species in the culture using the ribosomal genes. And also, we screen the ribosomal genes for bacteria, and we found that we have between 12 to 19 species, depending on the method, of bacteria genomes in the dataset. Unfortunately, most of these bacterial genomes are partial, they are not in a single contig, and also the species that are present in our culture are not present in public databases, which would help to clean the eukaryotic contigs. Also, from this primary assembly, we extract the mitochondrial genome. Monse's genome is a typical circular chromosome with a standard gene content as compared to the mitogenome of Thicamonas, the closest apusomonad reference. When aligned together, we see that they are also synthenic. They are structurally conserved, although Monse is a bit longer. Moving on with the pipeline to obtain the nuclear genome, we explored 4 different methods to classify the scaffolds or contigs into eukaryotic or prokaryotic. We used the unsupervised method called ESOM, uses tetramer frequency to create a self-organizing map that clusters together similar sequences. Then we also use MetaBat2, which also uses the coverage to automatically define bins. And then we move to supervised methods that use reference databases. We use the clean transcripts from Monse from a previous work. And we also use Tiara, a machine learning classification tool. Overall, we see that the four methods agree on classifying 390 scaffolds as eukaryotic. And currently, we're exploring the small differences to ensure we recover the best possible results. For example, we find that the eukaryotic ribosomal operon is in the RNA-seq and Tiara sets, but not in MetaBat or ESOM, because as expected, the composition of the sequence is much different than the rest of the gene. Then, we also observe that the RNA-seq is missing 52 contigs that MetaBat assigns as eukaryotic. This is also expected, since it is likely that the cells are not expressing all genes in culture conditions. And finally, the contigs recovered only from ESOM and Tiara do not have hits against eukaryotic protein databases. So we are still not sure if these contigs are relevant or not to get a good genome. Okay, so here I'm showing the statistics of the metagenomic assembled genome from one of the methods only, MetaBat, which is the most reliable one. So compared to the previous obtained transcripts, the draft MAC is quite complete and contiguous. Also, we can compare it with the reference Apusomona genome. Both genomes are similar in length, GC content, and Busco completeness. Our Monse genome is a bit more fragmented than the reference, having more and shorter reads though. This is why we hope that with the decontaminated reads, we can assemble a better genome using spades with careful parameters. In this table, I added the cleaner version of Monse's MAC. Again, being only from MetaBat, it seems that we are losing some genes, but we are increasing the GC content, reducing the number of scaffolds, and increasing the contiguity. So we think that combining with the other methods, we could retrieve a final version of this gene. And to sum up, we have extracted DNA from Monse in 3 different conditions. We have implemented a low complexity metagenomics pipeline that has successfully provided the second Apusomona genome. Although we probably need another round of decontamination with the 4 methods to proceed with the final annotation. Although Monse is freshwater and Ticamonas is marine, they both have similar nuclear genomes and mitochondrial genomes. But we're expecting to find more difference when we compare with the other species in this lineage. And with that, I want to conclude and thank you all for your attention.
Hello, everybody. Today I'm going to talk about the talk, which is Genomes of the Uncultured Marine Eukaryotes. They are needed, feasible, and useful. I start the talk saying why they are needed. In this slide, I present a few examples of marine protists, and we know that they vary widely in size. Some of them are very large from the microplankton fraction, some are very small. Large protists are well known for centuries. So these are the slides, images that were drawn by the microscopy existing at the end of the 19th century. So the groups of diatoms, dinoflagellates, Polycystenea, Acantharia, and ciliates are well known to be part of this large protist. On the other side, the smallest fraction of protists, the marine eukaryotes, which have sizes between 0.8 and 3 microns, they are very difficult to identify and to know which species do they belong. Even though they are widespread in seawater, they are very important and play important ecological roles as primary producers, predators, and parasites. I'm showing here an example of a study that we have been conducted in the Mediterranean Sea. We sampled monthly since 2001 And on the first graph, there is the temperature profiles during this more than 20 years. And in this sample, I have also been counting the number of heterotrophic and phototrophic marine picoeukaryotes, these very small protists. So in these samples, we have made an analysis of the diversity by a metabarcoding survey of 10 years, basically amplifying the before region of the 18S ribosomal DNA gene. And from these amplifieds, we just group the sequences in different amplicon sequence variants to see how many species are there and which is the temporal, seasonal, and interannual variation. In this first table, I show the number of different species that we found in different groups. And when we try to see how many of these species are already cultured, what we see is that except some groups, such as the mammaliophytes here and the chlorandrophytes here in white, the rest of the groups, they are present by a species which are very far from cultured species. So we are finding in a situation that we call it the uncultured majority, which means that the majority of microbial species, and that was known for bacteria, but also happens for picoeukaryotes, so the majority of these picoeukaryotic species are very different from cultured species. And basically 20% of the species, they do not have a match in a cultured species. And this is why, if we want to, there is a need to understand what these species do in the environment, and there is a need of have the genomes of these species, which still are not, we are not being able to culture. So having the genomes of these uncultured species is important because they are abundant, but I'm going to show here that it's also feasible to get the genomes of them. So the genomics from microbes normally depend on cultures, but as I said before, the dominant on the sea cannot be cultured. So how can we handle this? There is two ways to do it. I'm showing first one way, which is to get the single cell genomes from these uncultured species. And basically, we take a natural assemblage and we can sort cells one by one in the flow cytometer. We can conduct two different sorts. On the right is a sort of cells because they have plastids and they are pigmented. So the plastidic or phototrophic picoeukaryotes. And on the right hand, there is a sort of aplastidic cells which do not have pigment. So we can see them because we stain them. So we can sort one by one the cells in these two kind of populations. And then once we have one cell in a plate, we need to do a whole genome amplification cell by cell, which is a very critical step because it has to be extra clean, not to amplify contaminants on the plates on the lab. And once we have the whole genome amplified material, we can proceed to Illumina sequencing. We have done that in mild samples also from this Mediterranean site that I showed before. And I'm showing here the number of sacs of single amplified genomes that we obtained in the sort of plastidic cells, so phototrophs, and in the sort of aplastidic cells, so picoheterotrophs. And as you can see, each sort selects a different type of taxonomic groups. So some groups are only selected in the photosynthetics sort as primesophytes and mammaliophytes, and other groups like the mast, which are uncultured and known lineages in the ocean, and also some chlorandrophytes, they are sorted only in the heterotrophic sort. So we have this big collection of amplified genomes from different species, and then we can just take a look on several genomic properties. For instance, we can group, here there are more than 200 cells, 200 different sacs, and we can see which is the size of the genome that we get. In general, we get about, as a median, about 17 megabases for each, and with a big dispersion. We can also see which is the contiguity. So we have N15s about 15,000 bases, which is the completeness by comparing with the Busco databases. So we get about 40% completeness for each cell. And also we can take a look on how many genes are predicted from these sacs, which is about 10,000 cells, about 10,000 genes per sac. When we look at these properties, genomic properties, in terms of the different taxonomic groups that are retrieved, you can see this image. So the important part of this image also is to see that there are many taxonomic groups which sacs represented. Also to see that these taxonomic groups are the ones that are abundant in marine samples, and also to see that each taxonomic group has a different and reproducible genomic property. Because we are sampling many many cells from the same community, in some cases, the cells turn up to belong to the same species. And I'm showing here a few examples of that. So some more than 30 sacs were from species Minorisa minuta, and we can see as the genome, the completeness of each sac depends on the genome retrieval of each particular sac, meaning that there is like a linear relationship that if we can arrive to this linear relationship to 100%, then we can estimate the genome size of many different species that are present multiple times in our samples. And as you can see in this table, the predicted size is what is known and is expected for cells of this size between 20, 30, 40, in some cases up to 100 megabases per cell. The second way to get the genomes from the uncultured diversity is by metagenomics. And there was a recent paper that analyzed the massive amount of Tara Oceans data, the almost 800 metagenomes, and to retrieve marine amplified genomes from these metagenomes. So we published a paper saying that metagenome assembled genomes are for eukaryotes too, meaning that this approach that has been widely used for prokaryotes can be also used to retrieve the genomes from marine samples. The last part of this presentation, where I hope I convinced that there is a need to get the genomes of this uncultured diversity, and that getting the genomes now is possible, and by a culturing independent approach. And also, what do we do with once we have all this genomic data from cells, totally new genomes that didn't exist before? So I am showing you here only a few examples on how this can be exploited, this data. In the first example, obviously, is the most simple approach that would be to use this genomic data to participate in a better reconstruction of the eukaryotic tree of life. In this case, I'm showing a recent paper where they place, based on SAC information, on single amplified genomes information, they place the Picozoa, which were a group that were discovered almost 20 years ago and didn't have a good place in the eukaryotic tree of life. It placed this group in a very unexpected position, which is within the Archaeoplastida. And that was unexpected because Picozoa are known to lack chloroplast. So most of the Archaeoplastida groups obviously have chloroplast, but also it's the only example of a group that not only lacks chloroplast, but also it lacks any trace of having had a chloroplast. So it seems that it never had a chloroplast, and also it has to rethink about the evolutionary history of this Archaeoplastida supergroup. In another example, that was a published paper that we published 3 years ago, we found that many of uncultivated species of heterotrophic flagellates, and in particular in this case were belonging to the mast clades, which are clades that have been extensively found in marine waters. So we found that in the genomes of these species, we found the presence of a rhodopsin gene. Rhodopsins have been known to exist in many heterotrophic bacteria, also in some photosynthetic protists, but it was quite unexpected to find a pigment, a gene that uses the light on some heterotrophic flagellates. So that also opens a hypothesis of what this gene can be used in heterotrophic protists, but also coincides with the distribution of these species, which are very well known to predominate in the photic layer of the ocean. In the last example for the use of these novel genomes of uncultured species is that we can apply and use these genomes to recruit and extract the signal of molecular signal belonging to these species from large omics datasets. And in this example, I'm showing an incubation that we performed in the lab, where we incubated a natural assemblage in the dark for a few days. During this incubation, that we had metatranscriptomes during 6 days. During this incubation, the community changed, and we used the genomes to retrieve signal from the different species present in this mixed community. So in this individual graph is the number of molecular signal from this species during the incubation, And as you can see, the numbers, they follow different dynamics. And basically, the species which are in green here, which are photosynthetic species, they follow a dynamics of decreasing during the incubation. And this incubation was done in the dark, so that makes a lot of sense. While the species in yellow and in orange, in yellow, they were heterotrophic species, and in orange, they are mixotrophic, they increase it during this incubation. So once we have the signal, molecular signal from each individual species from a natural mixed community, then we can start to interpret the physiological function and activity of these species in the assemblage. To finalize this talk and to make the conclusions, basically the conclusions, I see that they are the title. So the genomes of uncultured marine pico-eukaryotes, I think they are needed because there is this uncultured majority. They are possible through single-cell genomics and metagenomics, and they are useful for many different applications in ecology and in evolution. And I end just making an appreciation of the people in my lab in this figure, but also people that has been very important in this single-cell work, as Oscar Fornas in Barcelona for the flow cytometry work, and Manuel Martín García from the University of Alicante for the whole genome amplification and David for the analysis of these cells. Thank you very much.
Hello, my name is Ori Francis. I'm an Excellence Fellow in the Blackster Lab in the Tree of Life program at the Wellcome Sanger Institute. And I'd like to talk to you about how more Protist genomes can help in silico prediction of eukaryotic complexomes. And by that, I mean predictions of proteins that form complexes across genomes, across the domain of eukaryotes. Now, a number of studies have explored how most of the eukaryotic diversity is distributed in the protists, that is, outside of the animals, the plants, and the fungi, even though we know that great diversity exists across the animal kingdom. This tree from Shannon Sibbald and the Archibald lab nicely shows how there are abundances and shortfalls of existing genomes and transcriptomes, particularly in some orphan lineages, conspicuous shortfalls there, but also how those lineages, the animals, plants, and fungi, are very narrow snapshots of eukaryotic genomic diversity. Now, Protist genomes are sparse largely because they are difficult to achieve, very difficult to acquire. So this is a chart from Genomes on a Tree, GOAT, and it really nicely shows how there are an abundance of animal genomes and animal entries, also corresponding to increased genome sizes. Now, it is the case that these reflect estimated genome sizes, but we do know that there are many protists with large genomes that at the moment are simply intractable to genome projects. And this is for a number of reasons to do with the coverage required, in addition to cobionts and surrounding bacteria for bactivorous protists. For more details, I do not have time to go into here. But it is a shame that we are yet to achieve these high quality genomes, because even with the Protist genomes that we have now, there are some incredible studies that can be done and some fascinating biology that can be unveiled, particularly about protein complexes, which again is my area of interest. And I have as an example here a figure from Jeremy Weidman and his work on the ER membrane complex, showing again like a complex, like a built up a complex repertoire of components for the last eukaryotic common ancestor, and endosome-related adapter complex 5 work from Joel Dax's lab, which shows again a novel adapter protein complex that again that wouldn't have been possible without the Protist genomes. So my question is, these in-depth studies, can we achieve automated exploration for searching protein complexes across the domain of eukaryotes? So I've embarked on this using a comprehensive comprehensive data set as a bait, and this is the EBI protein complex portal. This comprises around 6,500 queries, and it will go and it corresponds to ongoing expansion. So it's important to say that there are actually no Protist proteomes in the EBI complex portal, but what there are experimentally verified complexing proteins. So it's a really good data set to explore protein complexes across eukaryotes. And before embarking on this work, I would predict that there would be a high degree of conservation in as many of the genomes as I could search. I also would like to select a subset of Protist genomes for now to show that this is this analysis is viable. But obviously in the future, what I would like is for this analysis to be in place for as many high quality Protist genomes that fill the gaps of underrepresented lineages across the domain of eukaryotes coming from the Tree of Life program. And I'd like to use the Amoebi software from the Joel Dax lab in the University of Alberta, because I like how it automates forward and reverse searches and can be pulled apart. So to begin with, I'll use BLAST and Hammer, but in the future, I think I'll be able to use alternate search methods such as AlphaFold and FoldSeek. So for the purposes of this talk, I'll show conservation using Coulson plots, and these Coulson plots I have examples here, and what they are disks that are broken down into segments, and a disk corresponds to an entire protein complex. A segment corresponds to a component of that complex. At the top, the gray disk shows you the architecture of the given protein complex. Blue in the larger disks corresponds to the conservation of a component. White, which none of these have, corresponds to an absence of that component. And the number inside corresponds to the number of homologs found. For those who are interested in which were the most abundant components I found, here is a list of them. They may be useful potentially for as phylogenetic markers going forwards, but I'd just like to point those out. So as I say, there's a number of results, a number of hits to the roughly 6,000 proteins in the complex portal, just for simple BLAST and Hammer searches. And rather than attempting to show off what corresponds to about 1.5 million hits in remaining 4 minutes, I'd like to take a snapshot of complexes, each corresponding to one of the molecular systems, one of the 31 molecular systems known to be conserved in the last eukaryotic common ancestor. And what I hope you can appreciate from this snapshot of the results is that even though they are highly cherry-picked, there is a lot of blue here corresponding to a strong level of conservation right the way throughout the eukaryotes that I've chosen. I think this further supports the idea of a complex last eukaryotic common ancestor that has a lot of these systems. There are some conspicuous absences corresponding to, in particular, Giardia theta and Bigeloia natans, which would require additional work and maybe artifacts of the way I've done the analysis or the genome that I have. But there is also absences in Encephalitozoan cuniculi, and I believe that actually probably real, as the reduction and compaction is well known for microsporidia. So in my last couple of minutes, I'd like to point out a couple of examples of interesting things that have come from the analysis. I found conservation of a collagen type 1 trimer in Toxoplasma gondii that I could not find in any of the other AP complex. So I looked in. Admittedly, it's not a huge data set, but I was wondering, I still am wondering, what is it doing there? And I did see this one paper by Pereira et al., which suggests that collagenopathy is a part of the toxoplasmosis mouse model. One question, as sci-fi as it may be, is the origin of the collagen in this pathology. Could it be in some part of toxoplasma origin rather than of murine origin? A perhaps less sci-fi example is the gamma secretase complex conserved in Phytophthora infestans, and I point this out because I'm not the first person to have noticed it, but to suggest that this type of analysis, this exploratory analysis, can quite easily point these things out, as John Kay et al. Have suggested. Since it is in Phytophthora, the function is almost certainly not limited to the processing of brain polypeptide related to Alzheimer's disease. But again, this type of high throughput analysis can help us achieve a starting point for looking into the functions of some of these protein complexes. So I'd like to thank everybody in Team 301, all collaborators who have sent us samples, Henning and Sandra for maintaining the complex portal, and Joel Dax and Leo Barlow for their work developing Amoebae, and you for your attention. Thank you.
Hi, I'm Amjad. I'm a PhD student here in the Tree of Life, and I'll be telling you a little bit about the microsporidian genomes I've been generating and what they've told us about microsporidian biology. So, just to kind of begin, I really like this quote, and I really like the metaphor of Indra's net. And it's this idea that all sentient beings are interconnected with each other. And this is really how I think about biology. We may sequence a single organism, but actually that organism isn't just the target organism you're interested in, but it's filled with bacteria, it may have parasites, it may have different things living on it. And that's really what I do here with microsporidia. Just to kind of tell you a little bit about what they are, microsporidia are these intracellular, single-celled parasites with an extremely broad host range. They inhabit all of metazoans, probably, and even some protists. If you get them, they're really nasty. They cause diseases. And typically they've got this really fun infection or invasion mechanism where they've got this coiled loop called the polar filament. So the spores live everywhere. You ingest a spore, and the spore germinates by shooting out this polar filament, which looks almost like a harpoon structure. It pierces the host cell, and it injects the host cell with its own nucleus and some cytoplasm, and that cell then replicates inside the host. So just to kind of say again, there are more than 1,600 species currently with an extremely broad host range. They typically cause diseases. They're the causative agent for honeybee colony collapse, typically. And in Anopheles mosquitoes, they are a particular case where there are two species that don't really cause diseases in mosquitoes, but they seem to block malaria somehow. They seem to be associated with a decrease in Plasmodium transmission. So they may form a potential malaria control route in Anopheles, and they're of interest, at least in this manner. So this whole story really started with the Silk Road, and it was a strange story between silkworms, which are producing lots of silk, and Pasteur and Nageli, which you may recognize from milk. And the whole story is that at some point the silk yields were really decreasing, the silk quality was really poor, and what Pasteur had then discovered is that there were these microsporidian parasites inhabiting these worms. And the way they found that out was with his passion for microscopy. And this was how he devised a way to kind of deal with this problem. So just to go back again to what I do, which is really just pan for gold. So every time the Darwin Tree of Life sequences a genome, sequences an organism, I try and see if it's infected with a microsporidian parasite or not. And if it is, I then do some bioinformatics to generate that genome. And what I'll be telling you a little bit about is a few different stories that we've learned from those genomes. So just to start with, we've generated over 50 microsporidian genomes as of today. And this is a microsporidian tree, a phylogeny of microsporidia, and the different groups are highlighted in bubbles, and the tips in red symbolize genomes that I've been able to generate. And as you can see, they kind of scatter all over the tree, which is really, really exciting. One of the genomes that I've generated happens to come from a bark louse, really, really tiny organism. And this genome happened to have really high coverage, and it was a tetraploid, super exciting. And we happened to get some high-C data for it, which is chromatin capture data. It tells us how closely physically related sequences are. So you use it typically to scaffold sequences together to go from just contigs to chromosomes. And this is really what was done in this case. So we were able to build this into a chromosomal tetraploid. But what's super exciting about it is that if you look at this pattern closely, I know it looks a little bit like a rug, but what you can see is that each chromosome has 4 copies, and they seem to be arranged in pairs of physical similarity. So I think this will clarify a little bit further. So you can see that chromosome 1, for example, in its 4 copies forms 2 compartments, the green and the purple, which seem to be each physically associated together. So yeah, once you've looked at it a little bit, it becomes a little bit clearer. And this is not just a pattern that extends for chromosome 1, but it really happens for the whole genome. So chromosome 2 has the same thing, but not just that. Pairs of chromosomes can be arranged into 2 large genome clusters. So chromosome 1A, 1B, starting from the left here, are associated with chromosome 2B and 2C, whereas chromosome 1C and 1D are associated with chromosome 2A and 2D. And this is a pattern you really extend for the whole genome, which means that your genome, your tetraploid, is then arranged into 2 large genome-wide clusters, which are formed of diploids, so 2 diploids. The haploid is made of 10 chromosomes, around 17 MB each, but the whole genome can then be clustered into these 2 large clusters, and each cluster is 20 chromosomes. So then the golden question is, what could those 2 clusters be? And we hypothesize that the 2 clusters are probably the 2 nuclei that live in microsporidia. Some microsporidians are diplokaryotic, some are not, meaning that some have 2 nuclei and some do not. So we believe that each nucleus is a diploid in this case. So if the 2 compartments are indeed nuclei, how they are phylogenetically related can tell us a little bit about their biology. So there's a few mechanisms to kind of think about this. You may start with 2 nuclei, each of them are haploid. They then become diploid, each of them, and then they're just inherited independently, where you would expect this kind of phylogenetic clustering of copies AB and CD. They don't really like each other in terms of nuclear or sequence similarity. Whereas if you start with one diploid nucleus, you then become a tetraploid, i.e. you have two diploid nuclei, and those are still inherited independently. You wouldn't really expect some kind of phylogenetic clustering in this case. And the other option is that you have some kind of two diploid nuclei, they fuse, they recombine, and then they split again, i.e. the occurrence of recombination, sexual reproduction, and we still wouldn't expect phylogenetic clustering in this case. So I spent a long time building trees from the 4 copies of each chromosome and counting topologies. And what this told us is that there was no phylogenetic clustering at all. So another way to kind of think of this is that if you walk along chromosome 1A and then you ask which other copy of chromosome 1 is the most similar as you go along, you find this really strange mosaic pattern where if you start from 0, you'll see that the green copy seems to be the most similar, then the orange one, then the blue one, then the orange one again, then the blue one, then the green, and so on. So chromosome 1A is most similar to different homologous chromosomes at different positions, i.e. no phylogenetic clustering, which tells us that it could really be one of those 2 cases. And we really cannot distinguish this without population level data to really find out what is happening at the population level. So if you do have any tetraploid microsporidian samples, please do get in touch with me. So just another really story to walk into this is that we had published earlier in the year that polyploidy is widespread in microsporidia. We've seen it in many different species, and now we've been able to add a couple new leaves to this tree. And the species with an asterisk next to their name shows species that we've seen, that we've sequenced from DTOL. And as you can tell, the tree has grown. We've got new groups of microsporidia on here, and still the message remains that polyploidy is not just widespread in microsporidia, but ploidy seems to be dynamic across the tree. So the other thing I really wanted to mention is that some genomes are imperfect diploids or polyploids, meaning that if you look at a human genome, for example, you would expect that chromosome 1A and 1B are vaguely homologous to each other. They have the same gene order-ish. They look similar to each other, but this isn't the case in many microsporidians where although chromosome 1A, 1B, 1C, 1D have the same genes for the most part, they will have very large synteny breaks, lots of rearrangements between these copies. And if you were to build an assembly graph of these genomes, it would look something like this figure on the left, which is meant to represent a mandala, where cause and effect are really just absolutely intertwined, and you can't really make sense of one effect from one cause. And I've got a self-alignment of an unphased polyploid genome on the right, and you can really see these breaks in the dot plot, which symbolize these synteny breaks. So then the question, at least from the polyploidy literature, is that if you get these really large arrangements in a polyploid, it is typically thought that they may stem from the polyploid being an allopolyploid, i.e. it being the child of 2 different species or very divergent organisms. And the idea is that if you're a hybrid polyploid, there's gene dosage compensation issues, your genomes are very diverged, and this will, or you'll get erroneous homologous recombination, and this yields these rearrangements that we see. But this is actually not what we see. So what I've done here is I've kind of plotted the average divergence between the genome copies of a rearranged genome in green and a synthetic genome in purple, where all the 4 copies are very, very synthetic. And this is not what we see at all. We don't see a signal of divergence. We don't see this kind of clustering of 2 copies that prefer each other. So actually, it seems that both rearranged and synthetic polyploids are autopolyploids and not allopolyploids. So there may be a different explanation for why we see these rearrangements, but we're not there yet. And with that, I'd like to say thank you to my supervisors, Mara and Mark, and thank you for my colleagues at the Darwin Tree of Life. This work would not be possible at all without you. Thank you.
Well, thank you all. Amazing talks. It has been very interesting. I love to see Protist genomics talks because, I mean, they always reveal very cool and unexpected stuff. So now we have 10 minutes for a Q&A, more or less. And if you're okay with that, we are gonna start with the questions that we got in Slido through the live streaming. Okay. And I'm gonna ask you these questions by order of popularity, because in the system you can vote for the most popular one. So that's what I'm gonna be doing, okay? So the first question is from Mark Blackster, and it's for Ramon. So Ramon, have you tried supplementing the Illumina SAC single amplified genome data with nanopore or PacBio long reads?
Yeah, this is a very good idea, and we have not tried it. And I think we have very nice material to do it, because we have some SACs that at the Illumina level looks very interesting. So we have very good candidates to try that. So, but we have not yet done.
Okay, thank you, Ramon. The second question is for Guifre, and it's also from Mark. Guifre, have you tried training K-mer-based read binning algorithms on your initial sort of the assembled data into bacterial versus target eukaryote, and then resorting the primary read data?
Okay, thanks for the question. I'm not sure I understood the question, first of all, but we tried to separate the different reads by K-mers, classifying the eukaryotic reads versus bacterial reads using databases. But unfortunately, both eukaryotes and bacteria in our samples are so different from the species present in these databases that it was not easy to fully separate the different reads, if that was the question, of course. Yeah, I don't know if he can reply or anything.
Okay, well, I'm pretty sure that Mark will find a way to get back to you if that was not the right answer, or if he needs more information. Thank you, Guifre. So there's another question for you from Felipe Simeonato. I would like to know more about the Protist within opisthokonts. How diverse they are, where can they be found, which brother groups they share, brother sister.
I think it's quite a long answer for Felipe. I was thinking that I could suggest some papers or reviews on the matter for him or anybody interested to learn more about opisthokonts protists. But basically, we have between 3 or 4 lineages sister to the animal lineage, and we have also 2 or 3 lineages sister to fungi, and they can be found almost everywhere. The holozoans more in marine environments. Sisters to fungi are often from freshwater environments, but there has been a lot of moving around and different diversification within these groups. There's a lot of conversion evolution also, so it's a pretty interesting group of lineages. Yeah, I think in Slido I will just copy maybe some references, some literature. Okay, so thanks for asking.
Okay, thanks again, Guifre. There's a question from Amjad, for Amjad from Carmen Dinman. What is the most surprising or unique Darwin tree of life species you've found microsporidia sequences in?
Oh my God, hard to say, I think, to pick one. But I think the most surprising is a spider, because microsporidia have not been seen in spiders at all as of now. So now we know they're there. But I wouldn't know exactly the species without looking it up.
And that question comes from me now. What about the Epicomplexans? They are known to be parasites of gregarines, right? And so have you found any microsporidia unexpectedly in, I don't know, Toxo, Plasmodium?
So far, no. But I mostly things that are sequenced within Sanger happen to be arthropods are the things I've looked at the most, really. So I've seen some gregarines in arthropods, but I've not seen
gregarines or microsporidia in gregarines in an arthropod just yet, but I wouldn't be surprised if it happens.
That would be awesome. Okay, there's a reply by Mark Giffre regarding his previous question. Giffre, the idea is that the strong prior you get from within the data might aid in separating the cloud of unknowns. Okay, so just food for thought for your analysis. I have a question for Ori and Amjad. Based on what you have presented, how do you think having more Protist genomes available will change what we currently know of the rules of life? Because you are looking at things that, I mean, are novel, unexpected, that they don't follow what was supposed to be happening in the system based on the information that we currently have that is mostly based on model organisms, right? So how do you think the pictures of the rules of life that we currently know will be changed by exploring deeper the Protist genomes?
I think on a system, yeah, on a system, cellular system level, potentially it could upend a lot of what we see. I mean, the work that I've shown is homology to existing systems, existing complexes, existing proteins. But of course, in a lot of cases, there will be completely new innovations instead of homologs to those things that I'm looking for. So this is one of the reasons why it may not be as economically interesting to look at something that's free living and minding, you know, a heterotrophic protist that's minding its own business. But these things represent such a wide taxonomic group, such a break from the norm that we really should be enthused to go after these things. I don't know if you want to say more, Amjad.
Oh, I mean, I agree, absolutely. And I think we're just going to learn lots of new things. I mean, I've gotten to interact a little bit, at least, with reproduction in microsporidia. Now we have a slightly different idea of how things are evolving. Things like ploidy, for example. And I think we're just going to get a different sense of how things are actually happening. This will just be more data points, really, to add to our understanding. Exciting.
Okay, thank you. Thank you so much. So we still have, I will say, 2 more minutes. A final question for Ramon. So you are generating genomes from protists that we cannot get in culture. The rules are clear, or the guidelines are clear to define what a reference genome is within the Earth Biogenome Project. But what will be the characteristics of one of these single amplified genomes to become referential for the community working with protists? What would you require from these genomes? So not being partial, not being complete.
Yeah, by definition, the genomes that you will get from a single cell, since it has the amplification level, they are going to be in general partial. Perhaps they can have 20% or perhaps 40% of the genome. I think then it's quite interesting to combine several cells, several sacs from the same population, then you can complete the genome. So I think at the end you can get almost complete genomes from single cells, which I guess still probably the synteny, the contiguity to get to the chromosome level, probably it's not going to be possible, but still it will contain a huge amount of genetic use for many applications.
Thank you so much, Ramon. So there's less than a minute remaining, and I will close this symposium by thanking again the speakers. Thank you so much for your talks. They have been very insightful. And I would like to thank also all of you attending the symposium, watching the live stream. Thanks for being here. Thank you for your questions. And thanks to all the organizers of Biodiversity Genomics 2024. This is a great initiative. Keep doing this every year. Thank you so much. see around and sequence protists.